Let's now look at an application of DNA replication. The polymerase chain reaction, which its acronym is PCR, is a technique that mimics natural replication. In this technique, what we're going to have is that DNA double helix will unwind, and then DNA polymerase is going to be making copies of each strand of DNA that it is in the reaction because it will utilize it as a template. In PCR, since it is a chemical reaction, we need a container to place the reaction, and that is known as a PCR tube. When it comes to the different components of a PCR reaction, we have the DNA sample that needs to get amplified. The primers are going to be the segments of DNA that are going to guide the enzyme to amplify a specific section of the DNA sample. We need to build those newly synthesized strands, so we need the subunits. That's why we add nucleotides. We need an enzyme that is going to catalyze the reaction of bringing those nucleotides together, which we are going to utilize the enzyme DNA polymerase. And all of this needs to be placed in a particular solvent. This solvent is going to be a buffer system that it is particularly the correct pH for the DNA polymerase to actually catalyze the reaction. All of this reaction that is placed in the PCR tube is then placed in an instrument that is known as a thermal cycler, which is where the reaction will occur. Once the reaction is in the thermal cycler, there's going to be different stages that the thermal cycler will make that DNA undergo in order for the amplification of the new synthesized strands of DNA. The different stages or processes for the PCR reaction include a denaturing stage. And in the denaturing stage, what's going to happen is that the template DNA will separate. Typically, this happens around 95 degrees Celsius. Next, we have an annealing stage. In this annealing stage, the primers that were placed in the reaction will actually bind to the template DNA. This typically happens around 55 degrees Celsius. Next, we're going to have the extension stage. And in this extension stage, which happens around 72 degrees Celsius, what is going to happen is that the DNA polymerase, along with the nucleotides, are going to create and synthesize the new strands of DNA that needs to be amplified. PCR is a standard research technique for detecting mutations associated with genetic diseases. It can also be utilized to quickly detect the presence of unwanted DNA, as in the case of bacterial or viral infections. Lastly, PCR has also permitted DNA to be amplified from extinct mammals, Egyptian mummies, and even ancient insects trapped in amber. Now we're going to proceed to talk about the central dogma of molecular biology. The central dogma of molecular biology is a well-established process by which the genetic information that is stored in DNA is expressed in the structure of synthesized proteins. The genetic information contained in DNA is first transferred via the process of transcription into an RNA molecule. Then that transfer information that was stored as an RNA molecule, it is translated in order to create the structures of synthesized proteins. This whole process of transcription and translation is known as cellular gene expression. As we learned before, a gene is going to be a structure that contains DNA. And what we see illustrated in figure 13.1 is where does the process of transcription and translation occurs in an animal cell. As you can see, this area that it is in a circle that is lavender color is going to be the nucleus of an animal cell. In there, the process of transcription is going to transfer the genetic information from DNA molecule into what is called messenger RNA. We can observe here in this summarized version of these processes that overall in the nucleus, we're going to have the DNA that when that RNA molecule needs to be produced, the RNA polymerase is the enzyme that makes RNA. It produces a pre-mRNA that needs to be processed. And then overall, the end product of that is mRNA or mature RNA. That mature RNA is then shuttled into the cytoplasm. And specifically, enzymes that are called ribosomes are going to be utilizing the mRNA as a template in order to synthesize, as you can see in these red ribbons, the protein that results from that template of RNA molecule. Specifically, the process of translation is going to happen in the cytoplasm. Now let's focus on the other nucleic acid, which we know as ribonucleic acid, in other words, RNA. We learned previously that DNA is a double helix structure. RNA is an unbranched polymer consisting of nucleotides joined together by phosphodiester bonds. The length of RNA molecules can be as few as 73 nucleotides, but as many as thousands. And RNA, as I mentioned before, is the most abundant nucleic acid that exists in cells. When it comes to the particular way in which we have the molecule of RNA build, remember that at its basic nucleotide, what we have in RNA is going to be a sugar unit, that it is a ribose sugar. We're going to have a phosphate group. And in the case of RNA, we do not have thymine. Instead, uracil is what it is observed. I'm going to change the spelling of adenine here to be consistent with correct spelling for that nitrogenous base. Notice that many RNA molecules are actually single-stranded, which is one of the difference with DNA because DNA tends to be double-stranded. There are different types of RNA that exist in cells, and these include ribosomal RNA, in other words, rRNA. Understand that in cells, the majority of the RNA is going to be in the form of rRNA, because in the second column, you can see that 80% of the cell has this type of RNA. The other type of RNA is going to be transfer RNA. Transfer RNA is what delivers amino acids, which are the building blocks of proteins. Okay. Let me just quickly mention that ribosomal RNA constitutes about 65% of the material in ribosomes, which are the sites for protein synthesis. Lastly, the other type of RNA that exists is going to be messenger RNA. which, as you can see, out of the three different types of RNA, the one that is in the least amount is going to be the mRNA. mRNA, which sometimes is called messenger RNA or mature RNA, it is a carrier of genetic information from DNA that specifically it's the template for protein synthesis in a cell. And as I just mentioned, the protein synthesis, in other words, RNA translation, happens in the cytoplasm. Now, let's look at the structure out of those three, specifically of tRNA. tRNA is the molecule that it is important for protein synthesis because our tRNA is what brings the amino acids into the process of translation. tRNA have what is called an anticodon. And that anticodon is a three-base sequence in tRNA that is complementary to one of the codons in mRNA. Additionally, at the 3' end of a tRNA, that's where the amino acid binds, as I just mentioned. At the 3' end, that's where the amino acid binds. We can see that in figure 13.20, we have different figures just illustrating specifically the structure of tRNA. In the first one, we have a two-dimensional structure. Next, we have a three-dimensional structure, and then we have the simplified cartoon symbolizing a tRNA. Let's now talk about the process of transcription. And again, this is a simplified way to look at the process of transcription. What happens during the process of transcription is that first, the DNA double helix needs to unwind near the point where the gene needs to be transcribed. The process of transcription is mediated by an enzyme that is known as RNA polymerase. And RNA polymerase is going to read DNA in the 3' to 5' direction to synthesize the RNA molecule in the 5' to 3' direction. We earlier saw what 5' to 3' meant. And if we go back to slide 12 and we look at the chemistry, let's remind ourselves that that 5' to 3' direction that is building DNA molecules or RNA molecules simply refers to the positions of the carbon atom, which is 5' and its phosphate, and position 3 in its hydroxy group of the neighboring nucleotide. The chemistry is not any different when you're doing DNA replication or if you're doing transcription. The only difference is in the base pairing, as we're going to observe in a moment. Lastly, RNA is actually released and it rewinds to form double helix. So what happens is that in the last step of the transcription process, the RNA single-stranded is released, and then the molecule of DNA is going to basically rewind and reshape. Now, let's practice how to actually write what will be the RNA molecule that comes from a template of DNA. So What we're doing here is actually doing the process of transcription in a very small segment. So if we're trying to write what is the base pairing that occurs in transcription, every time we have an adenine, that is going to base pair with uracil. So if in the DNA we have a thymine, that is going to base pair with adenine. And then if you have a guanine, that is going to base pair with cytosine. And cytosine is going to base pair with guanine. So this is the base pairing that occurs. And pretty much what this is saying is that if this is the template, this is the new strand, which in this case is going to be specifically the synthesis of RNA. As I just mentioned, RNA is made in the 5' to 3' direction. So when I look at this example, every time I see a C in the newly synthesized strand, there's going to be a G. Now, different from complementary DNA, if I see an A, then that is going to base pair with the letter U. T is going to base pair with A, so I write these two A's. G is going to base pair with C, and then I finish up my answer. The complement of CCG is going to be GGC. And lastly, the complement of ATC is going to be UAG. And that's how you write what is the RNA molecule that could be synthesized from a template of DNA.